bonsoir public. Bernard Brais, je vous accueille aujourd'hui au nom des Instituts canadiens de recherche, l'Institut de génétique. C'est un café scientifique qui se déroule au Musée McCord dans le contexte du troisième symposium international sur l'ataxie de Charlevoix-Saguenay. Le but du symposium, c'est de dialoguer avec des gens qui font de la recherche, mais des gens aussi qui militent pour la recherche sur l'ataxie de Charlevoix-Saguenay et de placer ça dans un contexte beaucoup plus large. Ça veut dire de placer ces rechercheurs dans le contexte de la neurodégénération, donc le vieillissement prématuré du système nerveux et des neurones, surtout quand c'est relié à une dysfonction mitochondriale. Donc, on a invité ce soir pour la soirée 3 personnes qui sont très chères au projet. La première, c'est Sonia Gobeil. Bonsoir, Sonia. Bonsoir. Donc, Sonia est la co-fondatrice de la Fondation de l'ataxie de Charlevoix-Saguenay avec son mari Jean Groslot. Donc, on va lui poser quelques questions. On aura par la suite Elena Rugarli, qui vient de Cologne, qui est une Italienne qui vient à Cologne, qui travaille beaucoup justement sur certaines maladies mitochondriales qui ont aussi des manifestations ataxiques. Donc, elle s'est jointe au symposium pour nous partager son expérience, sa compréhension de ces pathologies-là. Alors, bonsoir. Et Heidi McBride, qui est notre chercheur local par excellence, chercheur de McGill et de l'Institut neurologique, qui est vraiment une des princesses du monde mitochondrial au monde. Et donc, j'ai la chance de l'avoir comme voisine, mais ce soir, vous avez la chance de l'entendre parler sur les mitochondries, ses idées un peu sur le lien entre les problèmes mitochondriaux et la neurodégénération, qui est le thème général. Donc, j'allais commencer par des questions avec Sonia pour nous mettre un peu dans le bain, qui n'est pas seulement de la science, mais aussi le côté humain de maladie. Et donc, Sonia, toi et ton mari, vous avez, il y a plusieurs années maintenant, commencé une fondation qui, à nos yeux, mais aux yeux de beaucoup, est un franc succès. Peut-être nous rappeler un peu les débuts et les pourquoi, et comment cette entreprise, cette aventure s'est développée sur le temps.
En fait, c'est une aventure qu'on n'a pas choisie. C'est une aventure qui nous est, disons, un petit peu tombée dessus par hasard. En 2006, on a eu un diagnostic pour nos enfants qu'ils étaient atteints de l'ataxie Charlevoix-Saguenay. Alors, c'était une grande nouvelle, un grand malheur, bien sûr. C'était une maladie dont moi, je suis originaire de Chicoutimi, mais dont je n'avais jamais entendu parler. Alors, ça a été un double choc. Mes parents sont âgés, vivants, et on est issus chacun de grandes familles. Et à nos connaissances, personne n'était atteint de cette maladie-là. Alors, on a fait ce que j'imagine tout le monde ferait à ma place. On a consulté le site web et là, on a cherché là-dessus. On a vu l'importance de la maladie au niveau de ses symptômes et de la dégénérescence. Et là, on s'est mis à regarder, bon, qu'est-ce qui se faisait au Québec, qu'est-ce qui se faisait à Montréal, qu'est-ce qui se faisait dans le monde, pour s'apercevoir que finalement, il n'y avait pas grand-chose. Alors, notre première idée, c'était de s'associer avec des grandes fondations qui peut-être pourraient chapeauter un petit peu la recherche sur l'ataxie Charlevoix, de faire un chèque, puis de financer, disons, via un don, la recherche. On cherchait sûrement pas à fonder une fondation, c'était loin de nos premières intentions parce qu'on avait nos enfants, on avait notre travail, c'était pas notre métier de faire ça. Et là, on s'est rendu compte, on a aussi, à travers tout ça, ça s'est un petit peu bousculé. On a rencontré les cliniciens au Québec qui ont les patients. Donc, on s'est déplacé à Québec, on s'est déplacé dans Charlevoix, on s'est déplacé au Saguenay et on a rencontré aussi un clinicien à Montréal pour savoir un petit peu qu'est-ce qu'il en était. Et finalement, on a réfléchi et après 6 mois, on a constitué un conseil d'administration. On a eu une réunion avec certains cliniciens qui nous ont tous dirigés vers Bernard Brais comme étant la personne à prendre charge de ce projet-là. Et de ce moment-là, en 2006, en juin 2006, on a fondé la Fondation. Et voilà. Donc, on a fait un commitment, on s'est commis à financer les projets de recherche qui évolueraient suite à la constitution de la Fondation. Alors, voilà. Et donc, l'objectif de la Fondation, bien, il y en a un seul, c'est de financer les projets qui pourraient amener un traitement, quoi qu'il est, là, à cette maladie.
Donc, dès le départ, c'était une façon pour vous s'associer à l'exercice de recherche, donc l'exercice de trouver une solution à la maladie de vos enfants. On se retrouve là, on est quand même plusieurs années plus tard. Aujourd'hui, on était 70 personnes à discuter ARSACS et des maladies reliées. Donc, les choses ont bougé. Êtes-vous, comme fondation, comme parents, trouvez-vous qu'il y a un immense chemin qui a été fait ou quelle est votre perception en général?
Oui, il y a une perception qu'il y a un immense travail qui a été fait parce que je pense qu'on est parti de rien, on est parti de scratch, comme on dit en bon québécois. Et puis, vous le dites, vous étiez 70 personnes, des gens qui se sont déplacés, qui sont tous occupés, qui travaillent déjà tous sur des projets scientifiques et qui se sont déplacés de Montréal, de l'étranger, du Saguenay, etc., pour être avec nous, en fait, à Montréal aujourd'hui. Donc, il y a des grandes avancées. Je suis un petit peu au courant de comment se font les pas, disons, presque à tous les mois. Je me tiens au courant, mais en gardant une certaine distance. Cependant, c'est jamais assez vite. On s'était donné le spectre du 10 ans. Je pense que c'est toujours ça qu'on voit dans les journaux quand on lit sur quelque chose. Ah, dans 10 ans, il y aura quelque chose. Donc, pour nous, c'était le 10 ans. Donc, je vous avertis tous, les gens de sciences dans la salle, il vous reste 2 ans. Donc, c'est jamais assez vite. Cependant, on demeure optimiste. Pour nous, c'est encore l'avenue qui était à envisager. Et puis, l'hypothèse de base, et qui est encore l'hypothèse, c'est qu'en ayant des gens qui travaillent sur une maladie quelconque, même si c'est une maladie rare, qu'il y a peu de personnes dans le monde qui sont affectées par la maladie, pour peut-être qu'ils pourront bénéficier dans le futur, les chercheurs qui travaillent là-dessus, sur l'ARSACS, par exemple, pourront bénéficier de percées, de connaissances, de nouvelles technologies qui est propre à d'autres plus grandes maladies plus communes. Et puis, nous, c'était comme ça qu'on voyait. Donc, d'avoir une équipe sur place qui travaille sur quelque chose pour nous, pour ça, mais en même temps, ces gens-là ont des yeux ouverts, puis il se passe plein de choses sur d'autres grandes maladies qui peuvent avoir finalement des similitudes. C'est ce qu'on souhaite, puis c'est ce qui, en tout cas, semble se décerner tranquillement quand je vous écoute parler de science.
Donc, c'est des grandes attentes, mais c'est des attentes qui, après toutes ces années-là, ont comme pris une sorte de chronologie un peu plus lente que vous le pensiez quand même. Mm-hmm. Et l'attente ultime, là, si on essayait de mettre ça comme dans quelques phrases, ce serait quoi? Qu'est-ce que tu dirais justement à nous tous chercheurs par rapport à vos attentes de parents? C'est quoi? Vos enfants sont quoi? Ils vont rentrer dans l'adolescence, ils sont certains. Donc, c'est quoi? Est-ce que tu as parlé de 2 ans? Vous avez parlé de 2 ans. Est-ce que c'est comme une attente qui est non dite, mais qui est quand même là en termes de chronologie?
Non. En fait, je ne mets pas je me prononcerai pas sur l'échéancier parce que j'ai pas d'idée. Je suis réaliste. Je sais que ça prend du temps. Je sais que c'est complexe. Je sais tout ça. Cependant, j'ai espoir. Je suis optimiste et puis je crois qu'éventuellement, il y a des choses qui vont pouvoir s'essayer, qui pourront améliorer. Je ne crois pas, je n'aime surtout pas le mot « cure » ou « traitement » ou « guérison ». Là, on prendra ce qui arrivera. Donc, même améliorer les symptômes, aussi petite amélioration qu'elle sera un exploit. Elle sera...
Formidable. Je peux vous dire qu'on vous remercie tous pour l'effort que vous faites pour vos enfants, mais tous les autres atteints. Et je pense que c'est un grand travail que vous faites avec beaucoup de style et de détermination. Maybe we should, because our focus today is science in a way, what are your expectations when you think of it? This is a foundation that backs clinical, basic science on ARSACS. What are the expectations of the foundation? Can you share that with us?
Well, we're expecting, of course, a treatment, but we're realistic on this. We're putting a lot of efforts. A lot of people are also putting a lot of efforts, the scientists themselves, into this work. So we know it's not easy. It takes time, but we remain optimistic, and we hope that even though it's a rare disease, that more common disease could eventually affect the the progression of the research on ARSACS, and links can be done, networks can be created, discussions. That's what we hope for. I have someone who has multiple sclerosis close to in my family, and he had something given on a drug for another disease, which didn't cure him, but helped him in his daily tasks. So I believe in that for sure. So, but we're not. We can't. We had the first view of having a 10-year. But after 10 years of funding and of research, we would eventually have something before us. But 2 years is the 2 years is still to be achieved, and I don't know. Maybe it will, maybe it won't. But I can't definitely. I don't know, and I don't think people working on the project exactly know when.
So we share that ignorance. So maybe we can share this the question of the need for research with our two colleagues. So I'll start with you, Elena. So you've been working for years now on mitochondrial basis of some ataxias and so forth. How do you see the field now and how it will evolve, and thinking very much in the perspective of the Foundation, which we're learning to treat, which is what they're doing, and I'm sure that's part of your motivation. Could you tell us more?
Yeah, I think what she was mentioning before is really very important. I think that what is at least my experience in this field is that we started to work, actually I started to work on a different neurological disease that was hereditary spastic paraplegia, and then it turned out that the gene we were studying, or the protein that is mutated in this disease, is interacting with another protein, and this protein is causing an ataxia phenotype, and this ataxia is very similar or shares some commonality to ARSACS. So I think that this the picture that is emerging is that is not really very good simply to focus on one individual pathway or one individual gene, that we have to try to put all of these into context and to understand what are the commonalities, because mechanisms can be in common, and this would be very attractive actually in terms of offering a therapy if we can identify that, and also interacting with other researchers or scientists that are studying more basic processes. For example, in our case, for a disease that involves mitochondria and neurodegeneration, basic mechanism of how mitochondrial function in a neuron, because this is at the end is why we need to understand why in all these cases we have mutations or lack of a protein that is present everywhere, but we have a very specific phenotype in the brain and specifically in a part of the brain. And so I think that interactions and try to understand better what are the basic mechanisms, how, for example, mitochondrial move in the neurons, or how the turnover of all the mitochondrial is achieved, for example, can be extremely important, together with understanding basic function of the individual proteins that are mutated.
So if we take it in the perspective of the Foundation, and it brings back to the comment of Sonia, is that there is overlap. There is obvious overlap, and maybe the treatment discovery will come through this overlap. So any ideas? Where are we going for overlap between other multiple neurodegenerative disorder and mitochondrial dysfunction and treatment for that? What's your perspective or your take on the issue of mitochondrial dysfunction and neurodegenerative disorder and finding treatment?
Yeah, this is actually it's a very challenging question.
That's why we're here. That's why we offered a drink before.
The answer to this question, I think it would be very, very nice. But I think mitochondrial dysfunction and disease is a large theme. And of course, when we think of mitochondrial dysfunction, we can, first of all, we should ask ourselves, is this an energy problem? That the traditional mitochondrial diseases are all considered diseases of respiratory chain, so the ability of the mitochondria to produce energy for the cell. With neurodegeneration, I think it's slightly different, because it's not always a pure respiratory deficiency, but what we have learned in the past is that other characteristic of mitochondria, like, for example, their ability to form a dynamic a network, so to interact with each other, to fuse and to divide, or to interact even with other organelles in the cells. And we have heard today a lot about ARSACS and possible interaction with, for example, the cytoskeleton. I have now the idea that these diseases are not really real, or let's say classical mitochondrial diseases, but that we have to develop sort of a different frame in which we have to see these diseases. And to think in terms of a future, in terms of a therapy, is maybe even more challenging in this case, because it's like the mitochondrial are organelles that are interacting with many other organelles in the cells, and we have to understand this picture maybe better.
So in a way, if I try to put that in the context of clinical teaching of a mitochondrial disorder, we're talking about mitochondrial dysfunction that are important or play a role in disease, but we're not talking about necessarily mitochondrial disorder. And I think that's the theme a bit of the symposium. Is ARSACS a mitochondrial disorder, or is there mitochondrial dysfunction? Okay. And if so, is it contributing to the disease? What's your feeling at this stage? I mean, you heard about the disease today. Are you still struggling with you haven't had all the information, but still?
Yeah, we're still. Yeah, I think that it's obvious that there is a mitochondrial phenotype in this disease. This is very obvious. That also in the mouse model has been presented today. There is this prominent transport defects of mitochondria. Could be that this is secondary to a primary function of the protein that is not related necessarily to mitochondria. But I think still that at least in many of these ataxia, mitochondrial dysfunction or certain role of mitochondria in these cells play an important, that mitochondria are important actors, let's say, in the pathology. So I wouldn't at this point discard completely the idea that in ARSACS there is a mitochondrial contribution.
No. Okay, good. So maybe it's a good transition to go to Heidi. Heidi has heard and discussed ARSACS for a while now, and at the same time you've been very interested in that dynamic concept of mitochondrial dynamism and function. General question. When we think of mitochondrial dynamics, your feeling at this point is why would the neurons be more affected and more involved with this process than other organs of the brain of the body? Why is the central nervous system and the neurons more vulnerable to mitochondrial dynamics problem?
Well, in some diseases that's the case, but in other diseases the muscles are the primary problem for mitochondria. So it's not so simple to say that, but it's certainly in a neuron the problem is that you have isolation. You're far distance away from the cell body. So the mitochondria drive out there, and then they're all by themselves. So they have to survive. So if there's any problem,
we all do.
There's more. They can't rely on their other neighbors so much. But I think the biggest thing about mitochondria that is both really exciting and really frustrating is the fact that there's such an old organelle that everybody feels comfortable with. That the last decade we've made amazing. Many people in this room have made fundamental new discoveries on how the energy powerhouse of the cell is what I say more than a powerhouse, right? It's doing all sorts of other biochemistry. They're dynamic. They're modulating calcium flux. They're doing so many things, and yet many of the neuroscientists don't really have them in their models, right? So metabolic stuff is not really studied all the time in neurotransmission. So there's a challenge for us to try and bring the basic level of our understanding of this organelle to a place where the assays and the tools are such that other people can start to adapt them in how they look at everything from memory to development to cell death, right? So I think from the I'm obviously, if this is a bench to bedside conversation, there's no question I'm really the bench person. But I think there's so much left to understand, and especially how mitochondrial quality control works, which is the big theme here. So, it's a combustion engine burning oxygen, and there's lots of damage being produced. And how do you remove that and make sure they stay healthy? And all those questions are hot in the field right now.
clearly hot, and that's one of the reasons why we wanted this symposium, is to be sure that for ARSACS in particular, that convergence of interest in mitochondria and neurodegeneration and ataxias in particular would be discussed, and the tree would be shaken as much as possible to see if other ideas would come out. How do you struggle with one thing that we were exposed today is the idea that you have a disease where clearly there's probably congenital abnormality, but it's really as the individual grows and continues to develop that something else happens, and as we progress, how do you see? Can you wrap your mind around that, thinking in mitochondrial
Well, the
terms?
mitochondriac people think, there's been this sort of theory of aging, which is based on mitochondrial dysfunction because they have their own genomes, and the idea has been, whether or not this is correct is still under debate, but mitochondria were from bacteria, so they have their own DNA, and this DNA is not repaired very well. So as the person ages, you can accumulate damage in the genomes, which will affect the electron transport chain, and you slowly accumulate dysfunctional organelles. And there's been long-term studies of sequencing of mitochondrial genomes that you do generate mutations, over age. So, but you hit on a point of, as I was sort of alluding to before, one of the biggest questions in the field is if you have, multiple patients with the same mutations, they can present very differently. So there's obviously external factors, other things going on here that is defining the presentation of the disease. So it's there's a lot of challenges, and I think we'll talk about that more tomorrow at the meeting in how can we make sense of this? It's for sure true, but again, given what we've learned in the last decade, if we can extend that into what we'll learn in the next decade, even at the fundamental behavioral aspects of mitochondria, I think that there's still many more things to understand. And the best thing about this kind of meeting is that it's unusual for the people like me to be hanging out with all these clinicians. I mean, it's not. That's why I came to the neuro, so I don't mean it's that unusual. But
you can say strange. It's all right.
No, but it's a great thing because it helps us direct what we're going to choose to do. It helps us define what the problems are in the clinic, and it helps us try and extrapolate what we're seeing into a physiological model system when we may just be in earlier model type behavior, cell-free systems or whatever, but trying to really bridge the gap and bring it up and make the steps work for translational research. And that's what has to be. We have to get much better at doing that, I think.
Maybe a technical question. I mean, this is for the Institute of Genetics, which has always promoted new technologies in genetics in particular. I think even in cell biology, there are revolutions that are often genetics. Do you think that at this point we're living a modification of tools that may affect greatly the consequences or the conclusions you're going to be able to draw from your work in the next few years? And if so, what are those technologies changing your world?
Oh, there's loads, and there's no question that the ability now to do high throughput interrogation of genes, of drugs, of model systems, of phenotypes. I mean, Richard's doing a bunch of stuff with drug discovery now in the Parkinson's field, and there's, I think 10 years ago you wouldn't find a basic scientist doing that kind of thing. So, in a lot of ways, it's really the wrong time to cut back funding for research, if I can just say that to CIHR. Because we're on like a log scale ability to find stuff, way more than we could before. It'd be single lab, single gene, single assay, and you'd publish and you'd get your grants and you'd be fine, right? But you wouldn't be doing this platform kind of research, and collaborating with all sorts of people and bringing people from everywhere all around, and the ability to push things forward through different model systems and through different mechanisms of interrogation, from exome sequencing to proteomics to lipidomics to like all the metabolomics, all of these things. The technology is just outstanding, and I think we have a lot to be excited about for the future.
So in a way, the optimism that Sonia manifested earlier is shared by you and probably by Elena too. I mean, we're living a transition technologically, better tools, better vision of overlapping fields. What you deplored a bit earlier was that the neuroscience scientific colleagues in your institution are still learning.
I still have to have a mitochondria in every slide, and then I'll be happy. But
yeah, so that's what we're trying today. In a way, the Café Scientifique was a way to bring mitochondria to the heart of everyone, including other people in the institution. So I think it's a good transition point to maybe open to the public. We have fantastic world experts on mitochondrial and ARSACS here. So if they would like, I would, out of politeness, ask them if they want to make a comment. Dr. Yule, you would like to put on tape a comment as maybe related to the conversations we had at this stage, and certainly Jean-Pierre will want to make a comment. So I'm going to just move the microphone, which is my role in life.
It should be a closure comment, but I cannot wait. Sonia, it is exactly 40 years ago that we started studying ataxias in Quebec with André Barbeau. That was in November 1974. And we, at that time, all the ataxias in Quebec and elsewhere in the world were called Friedreich ataxia. There was no difference, and everybody that was walking with some kind of difficulty, I think, was a Friedreich ataxia. So that Dr. Barbeau said we are going to do a study. That's 40 years ago, and he did really a big program, and applying that in Quebec City, we just realized that we had very little Friedreich ataxia, as we could see at that time. And I called him, and he said, yeah, that's interesting. You bring me your paper in 3 weeks. On that we were already in 1975, 76, and then that was the beginning of this big adventure, I would say, on ataxia. And I was, Dr. Barbeau died in 1986, and we have continued to work on ataxias and make other observation on other kind of ataxias in Quebec and elsewhere. But at 8 years, it's not long. 40, it's 5 times more. And I think we are all very dedicated to the study of ataxias in Quebec, and especially Charlevoix-Saguenay ataxia.
Question, comments? Yes, Dr. Yu? You can do the same if you want.
I just want to point out some of the promise in this field, the optimism I have. One is this is an autosomal recessive disease, likely loss of function. There are certain autosomal dominant diseases like Huntington's disease and Parkinson's disease with alpha-synuclein, and those have been very hard to understand because they're gain of function. So this altered protein, you get one copy, it's doing something bad, and none of them have been solved. But autosomal recessive, because you can't imagine what new thing could this gene be doing, or this protein. But autosomal recessive is more tractable because you just have to figure out what it normally does. And that gives you great insight into the etiology of the disease, and then you can think of treatments. And being an autosomal recessive, and I'm in the Parkinson's field, and there's 2 autosomal recessive mutations in 2 genes, and now they've been found, at least biochemically, to work in the same pathway, and it's related to mitochondria, so it's giving great insight. One other little comment is the technology is moving very fast, and another fortunate thing for this field is the mouse model. Like in the Parkinson's disease field, it was stymied. They discovered these genes, they knocked them out in mice, no phenotype. Pink one knockout mouse, no phenotype, it stymied the field for many years, and fortunately the Drosophila people broke it open when they mutated in the flies. And now you've got a phenotype of the mouse. I have obvious things to move on like tomorrow.
We're hiring you.
And then the third thing is the technology is, I think, there's this new technique where you can make gene knockouts. It's called CRISPR. And I don't know if everybody knows in the room, you know, the mouse in the Parkinson's disease has no phenotype. So you know what they've done in China? They've just knocked it out in pigs. And you couldn't ever do that in the old technology. So they've knocked out both alleles of Parkin in the pig, and we're applying that technology to map out pathways. So I think the time is right. And then the last little thing, because I did my PhD in botany, and I've been many years in a neurosurgery department. And I think it's really good to connect basic scientists and grow them up and communicate with the clinicians, because then we've got tools, we just don't know the problems. And when we live with the clinicians, we know the problems, and we can team up and bring different perspectives together. So, and this meeting is an example of the power of getting me and others more excited about ARSACS.
It's all a question of growth. Any growing questions from the public? Anyone would like to make a comment? Yes, Heather?
I would just like to extend what Richard said about the value of this being a recessive disorder. And I think there are some lessons we can learn from the study of muscular dystrophy, where, again, we have a very large protein. We didn't know it existed before. Yes, we figured out the function, but what are they doing in order to treat the disease? They're trying to find out the key parts of that protein that are the most important, and making mini genes and other types, and using other new genetic technologies to skip over mutations. So I think there's a lot of hope that when we look at other diseases, not only for mechanisms but for strategies of dealing with similar kinds of problems, even though the phenotype may be very different, I think we can get there.
Is there questions for the panel? I know we spent a long day already discussing many of those issues, and we'll come back to it. So, as I promised my panelists, I would not stretch it. I think I'm delighted that you came tonight and enjoyed a cocktail and this chat, and we'll call it an evening, but not completely, because they're students, and I gather we have to empty all the bottles, so do make a big effort. And thank you, you three. Thank you for the... And
Thank you very much.